Previously, we have seen that to genetically engineer an organism, you need to prepare a gene construct that contains a promoter, gene of interest, a reporter gene, and a terminator. Here we will see the most commonly used biological method to insert the gene construct in the organism we wish to transform, which uses Agrobacterium tumefaciens as a vector. Agrobacterium tumefaciens is a pathogen bacterium living in the soil. When the roots of a plant are damaged, their cells emit molecules which attract mobile agrobacteria from the soil. In addition to its bacterial chromosome, Agrobacterium also has a small, circular DNA called the T plasmid, T standing for tumor-inducing. The T plasmid has two major components which confer the bacterium its virulence: the Vir genes and the tDNA region. The Vir genes are responsible for copying the tDNA, transferring it to the plant cell, and inserting it in the plant genome. Once inserted, the tDNA is transcribed by the plant's own RNA polymerase. The tDNA genes code for the production of auxin and cytokinin, two hormones that induce uncontrolled growth and division of the plant cell, followed by the production of opin, which is a source of food for the bacterium. So Agrobacterium engineers plant cells to produce its own food, which results in the plant gall that not only looks ugly, but it also prevents the normal circulation of sap in the plant. Because of its capacity to integrate foreign DNA fragments into the plant genome and thus induce permanent transformation of the plant cell, Agrobacterium has been used as a natural genetic engineer since its discovery in the 1980s. To do this, the T plasmid is genetically modified. The infectious genes are removed and replaced with a synthetic gene construct. The only thing that remains intact of the tDNA are its bordering sequences, which are important. When an engineered bacterium comes in contact with a plant cell, the insertion of the tDNA will occur as usual. However, because the tDNA does not contain its original genes, after its insertion in the genome, the cell will express a desired trait of interest. This transformation with a biological vector has a higher success than the mechanical transformations, as the engineered DNA construct is directly inserted in the plant genome thanks to the Agrobacterium infection mechanism. Despite its relatively high success rate, genetic engineering of crops with Agrobacterium entails its own risk. The main inconvenience is that the insertion of the tDNA occurs at random in the genome, as we will see now in detail. The tDNA is not transferred in the cell on its own. It is followed by two enzymes, a ligase and a restriction enzyme, which are produced by the Vir gene. When the enzymes and tDNA reach the plant cell nucleus, the restriction enzyme will search for a specific, short sequence of DNA called a restriction site. When the enzyme finds this sequence, it binds to it and cuts the DNA, leaving two short open-ended DNA sequences. This is where the boarding sequence of the tDNA comes in play. They are complementary to the open ends of the plant DNA and will pair up. Then the ligase stitches together the tDNA and plant DNA by repairing the covalent bonds between their nucleotides. The issue with this method is that the restriction sites are short sequences which are not unique. In fact, there are thousands of these sites scattered around the genome. If there is a restriction site in the middle of a gene essential for the cell growth, cutting the plant DNA at this exact site will disrupt the gene expression, which can have fatal consequences for the plant cell. Plant transformation with Agrobacterium is rarely done by simply adding Agrobacterium in the soil of potted plants. More often, it is done using the tissue culture technique. First, plant cells are isolated and stripped of their cell walls to produce protoplasts. Then, a suspension of genetically engineered agrobacteria is added. The whole mix is incubated in specific regulated conditions until the cells are successfully transformed. Transformed cells are then grown in plants using the plant tissue culture technique. To avoid the step of plant tissue culture, the floral dip technique can be used. The flowers or floral buds are dipped in a suspension of genetically engineered agrobacteria, resulting in the transformation of the plant germ cells. These cells can then be used to produce seeds that will be grown in a genetically modified plant. An example of Agrobacterium transformed crop is the decaffeinated coffee tree. The gene of interest in the DNA construct contains a sequence coding for a small interference RNA. This siRNA recognizes a messenger RNA for theobromine synthase, which is translated into an enzyme that methylates xanthosines into caffeine. The siRNA binds with the theobromine synthase messenger RNA, effectively preventing its translation into a functional enzyme. Such engineered coffee plants can have up to 70% less caffeine than non-engineered plants, without alterations of the flavor of the beans. Moreover, a successfully transformed plant can be obtained within a year, which is a significant improvement compared to the average duration of coffee breeding programs that last for about 25 years. Another genetically engineered horticultural plant is the blue chrysanthemum. By inserting the flavonoid 3', 5' hydroxylase gene in the chrysanthemum genome, the production of blue anthocyanin is increased by 60% as the result of the activity of this enzyme. This produces blue flowers.